Now a little bit about endosperm. Endosperm or primary endosperm cells are formed within the ovule, within the embryo sac, after the fusion of male gamete with the polar nuclei, resulting in a triploid endosperm tissue. What happens to this endosperm tissue? The endosperm which is formed will divide. Endosperm cells which is having triploid nucleus will divide in such a way that there will be lots of free nuclei initially. Free nuclei. This condition can be seen in case of coconut. In coconut, the liquid part of the coconut is actually the free nuclei. It is actually the liquid endosperm. And as you know already that endosperm is filled with food reserves for the growing embryo. So this is what we see inside the coconut. Inside the coconut, what liquid part is there? The liquid endosperm is made up of free nuclei. So initially there will be free nuclei. After that, each of these nuclei will be surrounded by cell walls. And what we see in the white kernel on the sides, the white part, solid part of the coconut, what we see, that is the solid part of the endosperm. That is because cell walls are laid around each of these nuclei. The embryo will later on develop, and the embryo will derive the nourishment from these endosperm. Now about some of the seeds. Some of the seeds, as they mature, the endosperm will be completely utilized. For example, in case of pea, groundnut, beans, there will be no endosperm remaining in a mature seed. Whereas in case of castor and coconut, endosperm remains in mature seeds too. So we are talking about mature seeds here. In a mature seeds, in examples like pea, groundnut and beans, no endosperm remains after the seed matures because it will be completely utilized by the embryo. Whereas in case of castor and coconut, the endosperm remains in the mature seeds.